Of course, it will be very interesting to investigate the activity of the single neuron during the decision-making process. It is quite difficult task because neurons are quite small objects. So the typical size of the neuron is between 10–50 micrometers. So here you see on this graph the very small object compared to the tip of the pencil. So how to investigate the activity of this very small neuron? We can of course visualize the structure of this neuron using, for example, the electronic microscopy. But we also can put a small sensor, small electrode near this neuron or inside of this neuron and record the activity of the single neuron. So as you remember, the neuron is the main computational element of our brain. So dendrites, they accumulate information from other neurons. The cell body integrates this information and produces the action potential. And action potential propagates the axon and sends the signal to other neurons or to our muscles. So we can record the activity of one neuron using single cell recording method. So we put a very small electrode inside of the neuron or nearby, and we can record the single action potentials produced by the neuron. The more active the neuron is, the higher number, the higher frequency of the action potentials is produced by the neuron. So overall, the neuron is a major computational element of the nervous system. So neuron accumulates information, neuron is activated by other neurons or by the sensory organs. And if this activation exceeds a certain threshold, the action potential is produced, and the action potential propagates the axon and activate by the release of the neuromediators, the muscles or the next neurons. So how can we study the neuronal activity during the decision-making process? We can implant an electrode to the brain of the animal and train the animal to perform certain tasks, or for example, to pay attention to the screen. So I just give you a few examples of the reactions of different neurons to certain stimulus. So the presentation of the stimulus is indicated by the black line in this graph. So you see that different neurons react very differently to the same object. Each line, vertical line here, represents one spike, one action potential. So you clearly see that the first neuron produces a lot of spikes, a lot of action potential during the presentation of the stimulus. The second neuron actually reacts to the offset of the stimulus, and the third neuron reacts to the onset and to the offset of the neurons. On the right side of this picture, we represent the same data slightly differently. So we make a histogram, we calculate the firing rate, the number of spikes per second. And we will use these graphs quite a lot during the next lecture. So on these graphs, we clearly see that neurons are activated by the stimulus, and the first neuron is active during the presentation of the stimulus, the second neuron is activated by the offset of the stimulus, and the third neuron is activated both by the onset and by the offset of the stimulus. So during the course, you will see this kind of pictures. So here, monkey has to get food from the food box. You see the representation of the activity of the neuron, and each dot here represents a spike. So each line represents a trial. So what do you see here? That this neuron is particularly active when monkey touches the reward. So this neuron reacts to something that has a value. We can visualize this reaction of the neuron using a histogram. We can calculate the firing rate, the number of spikes per second. So you clearly see that when monkey touches something that has a value, these neurons are activated. So using this method, we can record the activity of the single neuron or the activity of few neurons at the same time. This is a very important method because we can directly measure the neuronal activity here. That's the only method that helps us to do it. Unfortunately, this method is invasive, so we damage a brain when we implant the electrode inside of the brain. Normally, this method is limited by few neurons. Unfortunately, we cannot measure the activity of billions of neurons at the same time. And to be honest, we do not clearly understand the informational code. So we don't know all details about the code, how information is encoded in the neuronal activity. But this method brings us unique insights into the decision-making process. We can also use EEG and MEG to study the activity of large portions of the human brain. So electroencephalography is sensitive for the electrical activity produced by the firing of neurons, and magnetoencephalography is sensitive to the magnetic fields produced by active neurons. So the neurons in our cortex are highly organized. So large groups of neurons can be seen as dipoles, negatively charged on top and positively charged in the middle. So this difference induces local electrical fields that can be recorded even outside of the brain using EEG sensors, EEG electrodes. So EEG method is sensitive to the electrical local currents induced by active neurons, by large groups of neurons. So these local electrical currents also induce magnetic fields, and these magnetic fields can be detected by the MEG method. Some of you perhaps are familiar with the EEG method. Using EEG cap, we can record EEG signal, and it looks a bit noisy. It is difficult, to be honest, to recognize something here related, for example, to decision-making process. So we can use some tricks, and we can use the technique called event-related potentials methods. So, for example, we can present sound or a visual stimulus to the person many times. So if we average EEG during the presentation of the same stimulus, we can filter out noise, and we actually get the signal related to the presentation of the stimulus. So in this case, you see the evoked response triggered by the presentation of the auditory stimulus. So this is the activity of the brain related to certain stimulus and, for example, related to certain stage of decision-making process. So we can use event-related potential technique to investigate decision-making process. Imagine that two subjects play the matching penny game. This game is played between two players, player A and player B. Each player has a penny. If penny match both heads or both tails, player A keeps both pennies. If the pennies do not match, player B keeps both pennies. We can record evoked responses to the outcome of this game. So in this case, you see evoked responses to the losses and to the wins during the matching penny game. So pay attention to the loss situation, when person lose money. You see a peak around 250 milliseconds, and this peak is particularly strong when person change decision in the next trial. So I visualize this effect on the right side of this graph. So you see that the peak is particularly strong when subject changes the decision in the next trial. So we can predict the decision based on the evoked response, based on the event-related potential. This method helps us to really investigate the dynamic of the decision-making process with the millisecond precision. We can also record activity of the brain using MEG, and MEG measures the magnetic fields produced by the active neurons, actually produced by the electrical activity of these neurons. And MEG uses extremely sensitive devices called superconducting quantum interface devices. This is a quite expensive method, but it also helps us to detect activity of certain cortical neurons during the decision-making process. It is important to remember that MEG and EEG do not reflect the action potentials. MEG and EEG reflect postsynaptic potentials. So with these methods, we actually monitor the activation of the neuronal populations by other neurons. So overall, MEG and EEG methods are strongly recommended if you would like to study different stages of decision making. Both methods have excellent time resolution. EEG method is quite cheap, but MEG device is relatively expensive. Unfortunately, both methods do not have the optimal spatial resolutions, but both methods are becoming more and more popular in neuroeconomics.